So this is the second to last video for all of cellular respiration. This should be watched for Friday. This is the last big chunk of what happens in cellular respiration. It's called oxidative phosphorylation. It basically involves two steps: electron transport and chemiosmosis. Keep in mind that the um, NADH and the FADH made from glycolysis and from the Krebs cycle are now going to be put into the last step to make the largest amount of ATP. The ATP that's created from glycolysis and from the Krebs cycle, they go off and are used for whatever other process they are needed for, but the NADH from glycolysis and the NADH and the FADH from the Krebs cycle are going to be used to create a very large amount of ATP. Then NAD, FAD, NAD are returned to be used again. So they're recycled. So chemiosmosis. The electron transfer in the electron transport chain causes proteins to pump hydrogens from the mitochondrial matrix into the intermembrane space. Basically, what's going to happen is that the hydrogen ions, when they diffuse back across the membrane, they are going to pass through ATP synthase, which we know is the enzyme that makes ATP. ATP synthase uses that flow of hydrogen to drive the phosphorylation of ADP to make ATP. This is just an example of chemiosmosis, or the use of a hydrogen ion gradient to drive some sort of process. So chemiosmosis is not specific to cellular respiration. It can happen in a variety of settings, one of them being the creation of ATP. If we look at this pump, the inside is going to be my mitochondria, the yellow, the orange is my intermembrane space. Basically what happens is I pump out a bunch of hydrogens, my little white balls, and they are going to basically create some pressure because I have a high concentration in one area. It's going to start spinning the ATP synthase, which is my light purple thing, and it's going to attach phosphate to ADP, and it'll make my ATP. Think about it this, and I just keep doing this. We want to think about, where do those hydrogens come from? They come from my NADH, my FADH, all of that's coming from the first two processes, donates all of their hydrogens to fuel to spin this pump that's going to make a ton of ATP. Again, we look at, you see at the base, a NADH, FADH. Basically, the first step is the electron transport chain. Basically, electrons get really excited and the hydrogens break off. As they are breaking off, they're building up that gradient. So we have hydrogens coming from NADH, FADH. We also have some hydrogens coming from water. And so the electron transport chain basically excites them until they break off. And then during chemiosmosis, all of those hydrogens get pumped through ATP synthase. It spins and it makes ATP from ADP and the phosphate. That energy that's stored in the hydrogen gradient couples with the reaction of ADP and phosphate, and it makes ATP or creates ATP. That gradient, it establishes what we call a proton motive force. Basically, think about it this way. If I have, it works the same way that like a dam works to harness energy, right? If we look at some dams, they'll stop the flow of water and pressure builds up, right? Because water is waiting on one side. If I open a very large hole, the water will rush out really quickly and then it'll relieve all of the pressure. Whereas if I have a tiny hole in my dam, the water is going to spurt through at a very high rate with a lot of energy, and that's how a dam harnesses that energy. And so similar to that, when I have all of my hydrogens in one place and they're pumped through a very small channel, that creates energy that can be harnessed to create ATP. Pause this, see if you can answer this question. If not, bring it up on Friday. Again, pause this, see if you can answer this also. Again, if we look, oh, my head's kind of in the way. So this is the summarizing slide for all of this. I get 2 ATP from glycolysis and 2 NADHs. I get 2 ATP from my citric or my Krebs cycle. I also make NADH and FADH from this. During oxidative phosphorylation, that NADH and the FADH from both of the first 2 steps are utilized to create from 26 to 28 ATP. Usually we say that we make 32 ATP for each cycle of cellular respiration. That's the total output. And so I only had to put in, if you think back to the very beginning, I only had to put in 2 ATP to get 32 ATP out. That's why they say like 30 to 32, because they count those first 2 as being not in the total. And so that is the end of that cellular respiration process. This video should be watched for Friday. There's one other that will be part of it.